From the ORNL website, Bringing Custom Microbes to the Business of Recycling Plastic. Link in the description below. Current plastic recycling methods require sorting by chemical composition, a method that is expensive and results in products that are of lower quality and value than the starting plastic. If plastic waste could instead be converted to valuable chemical intermediates, then economical use of mixed waste as a feedstock might be feasible. Scientists working on a solution for plastic waste have developed a two-step chemical and biological process to break down and upcycle mixed plastics into valuable bioproducts. Sullivan developed a two-stage oxidation and biological funneling approach that can break down and reform mixtures of common consumer plastics. The end products can be adjusted by metabolic engineering of the microbes in the second step, which should enable tailored conversion into various platform or specialty chemicals. The project, which involves multiple institutions, draws on synthetic biology expertise at the Department of Energy's Oak Ridge National Laboratory to engineer a microbe that converts deconstructed plastic waste into building blocks for next-generation materials. The new process, described in the Journal of Science, would replace a system that now requires painstaking, costly sorting of materials, which has resulted in only about 5% of plastics being recycled in the United States. Mixed plastics waste represents an abundant and largely untapped feedstock for the production of valuable products. The chemical diversity and complexity of these materials, however, present major barriers to realizing this opportunity. In this work, it is shown that metal-catalyzed oxidation depolymerizes commingled polymers into a mixture of oxygenated small molecules that are advantaged substrates for biological conversion. We engineer a robust soil bacterium, Pseudomonas putida, to funnel these oxygenated compounds into a single exemplary chemical product, either beta-ketoadipate or polyhydroxyalkanoates. This hybrid process establishes a strategy for the selective conversion of mixed plastic waste into useful chemical products. The project is led by the National Renewable Energy Laboratory and also brings together scientists from the Massachusetts Institute of Technology, the University of Wisconsin-Madison, and ORNL under the Bio-Optimized Technologies to Keep Thermoplastics Out of Landfills and the Environment, or BOTTLE Consortium. Different plastics contain different polymers, each with unique chemical building blocks. The BOTTLE researchers developed a process to convert mixed plastics to a single chemical product, working toward a solution that would allow recyclers to skip sorting. The first step in the process relies on oxygen and catalysts to break down large polymer molecules into their smaller chemical building blocks. The process was applied to a mixture of three common plastics: polystyrene or PS, used in disposable coffee cups; polyethylene terephthalate or PET, used in single-use beverage bottles, polyester clothing and carpets; and high-density polyethylene or HDPE, used in many common consumer plastics and often associated with milk jugs. This is a potential entry point into processing plastics that cannot be recycled at all today, said Greg Beckham, a senior research fellow at NREL and head of BOTTLE. The oxidation process breaks down these plastics into a complex mixture of chemical compounds, including benzoic acid, terephthalic acid, and dicarboxylic acids, that would require advanced and costly separations to yield pure products. That is where biology comes into play. BOTTLE colleagues engineered a soil microbe, Pseudomonas putida, to biologically convert or funnel the mixture of small molecule intermediates to single products, either polyhydroxyalkanoates or PHAs, which are emerging form of biodegradable bioplastics, or beta-ketoadipate, which can be used to make new performance advantage nylon materials. The experiment built on a process developed by ORNL's Adam Gus and colleagues at NREL to engineer the bacterium with desired traits from other organisms. The process, outlined in the journal Metabolic Engineering, converted deconstructed PET into building blocks for a superior nylon product that is more water and heat resistant, ideal for applications such as automotive parts. Experimenters took a combinatorial approach to pathway assembly, basically finding the best combination of genes from different organisms that allowed them to get a robust utilization of PET in Pseudomonas petita. Gus said, ORNL specializes in modifying non-model microbes to add traits useful for biotechnology, tapping into their deep expertise in synthetic biology as well as transcriptomics and proteomics to discover new metabolic pathways. Combinatorial chemistry is based on the principle of making many chemical compounds rapidly on a small scale in small reaction cells. Biological funneling simply means that we've engineered the metabolic network of a microbe to direct the carbon from a large number of substrates to a single product, said NREL's Alison Werner, a co-author on the science paper. To do this, we take DNA from nature, usually other microbes, and paste it into Pseudomonas petita's genome. The DNA is transcribed into RNA, which in turn is translated into proteins that perform diverse biochemical transformations, forming a new metabolic network and ultimately enabling us to capture more carbon and to tune where it goes. Gus and colleagues have spent years perfecting P. petita to convert the plant biopolymer lignin, derived from bioenergy crops, into advanced bioproducts as part of the Department of Energy's Center for Bioenergy Innovation and Agile Biofoundry. In 2020, Gus led a team that announced it had engineered the microbe to simultaneously digest 5 of the most abundant compounds of lignocellulosic biomass. In the next steps for bottle, we're continuing to expand the range of molecules that P. petita can eat as we work to break down more types of plastics and also more real-world plastics that have additional additives, Gus said. Plastics are major environmental pollutants and are largely made using fossil carbon, he said. This research sits at that intersection of breaking down today's plastic waste and converting it into building blocks for the next generation of plastics that are both recyclable by design and biodegradable. Funding was provided by Department of Energy's Advanced Manufacturing Office and Bioenergy Technologies Office. The work was performed as part of the Bottle Consortium. UT Batil manages ORNL for the Department of Energy's Office of Science, the single largest supporter of basic research into the physical sciences in the United States. The Office of Science is working to address some of the most pressing challenges of our time. For more information, please visit energy.gov/science.